Hello and welcome to OSDR Chats. Today we have the opportunity to speak with Professor Prof Borg of the University of Malta. While it's becoming well documented that spaceflight affects biological processes, not all pieces are well represented in current literature. Professor Borg and his team are filling in gaps in the understanding of the effect of spaceflight on hemoglobin formation and the implications on future astronaut missions. In this interview, Professor Borg shares his research with us and describes how the open science data systems and the analysis working groups contributed to this publication. Thanks for joining us today, Professor Borg. Could you please highlight the most important finding in this publication?
So thank you very much for the opportunity. It's indeed a pleasure and an honor for me. So in our study that we published in Nature Communications, so it's a study entitled The Spatiotemporal Expression and Control of Globin Genes in Space. So one of the most significant findings was the discovery that there are actual changes in the hemoglobin genes themselves, in the expression pattern. So we all know about fetal hemoglobin present in fetal blood and fetal life, and the expression of adult globins, which are of course the predominant globins in adults. And we found that when we tested and analyzed blood samples and data from NASA twins, Inspiration4 and JAXA, we found a perturbed globin profile that in space there is preferential expression of the fetal type of erythropoiesis or fetal type of expression. And concomitantly, the adult globin genes were sort of repressed or silenced. So that was a very, very interesting and significant observation that we did in our study. So of course, we also discovered that after spaceflight, so when the astronauts come back to Earth, these globin genes, especially the adults, so the alpha globin and beta globin, they showed upregulation to pre-flight levels even beyond. So this would indicate a potential compensatory mechanism during post-flight recovery. So that to us was pretty significant.
That's pretty fascinating. And my second question is, how do the open science data systems or the analysis working groups help assist you in your work so you could achieve these research goals?
Yeah, so without any shadow of doubt, I mean, these repositories and the databases, I mean, they play a very indispensable role, right? Because they facilitate, they enable us to access the data. And basically by providing this type of multi-omic data sets from key studies like these NASA twins, JAXA and Inspiration4, I mean, myself and my team, we were able to access this data, to integrate this data across multiple levels, across the gene expression. And of course, this includes the DNA, the RNA, and also protein levels. And this integration from the repository, from the OSDR, this was essential to map the spatiotemporal regulation of the globin genes that we found. So in particular, two data sets that we made use of, the OSDR 530 and the OSD 570. So this allowed us to observe in greater detail and quantify the expression of the said globin genes. So basically, this data collection facilitated by the OSDR, this combined huge effort by the AWGs, the analysis working groups across multiple groups. And this provides a database. This provides like a platform, right, for interdisciplinary collaborative effort, data sharing. I would dare say that without these OSDR data sets, I mean, it would have been so much more challenging to conduct the study to achieve these results. So the OSDR played a very pivotal role in facilitating these data sets.
I'm super glad to hear that you were able to do this research through the OSDR and the AWGs. Thank you for sharing your time and your insights with us and with our viewers.
Thank you very much.
And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access the datasets analyzed in this work with the link provided in the description section below. Thank you and have a wonderful day.